Welcome back to this next video in which we are discussing HPVs and cervical cancer. Okay, so in the previous video we went over background anatomy and physiology of the cervix. In this video we're now going to turn our attention to looking at background pathology, and the background pathology that I want to make sure everyone is familiar with and has at the forefront of their mind before we actually specifically discuss HPVs and cervical cancer is neoplasia, which is the process of how you go from having a normal cell to having loads of cancerous cells. How does a normal cell over decades give rise to cancerous cells? Where does cancer come from? So, let me just draw a little picture of this. So, in pink here, let's let this be a normal cell. So here is its nucleus here. So this is going to be a normal cell. And what can happen over years and years and years in a process that we want to understand completely is that this normal cell can give rise to loads and loads of cancerous cells. So what colour should I have the cancerous cells in? I think I'll go for some evil looking red colour here. Okay, so this is going to be our cancerous cell. Here's its nucleus. Okay, and of course a single normal cell will not just give rise to a single cancerous cell, it will give rise to lots of cancerous cells. So I'll put cancerous cells here. So this is the process that we want to understand. How does a normal cell end up producing a cancerous tumour, a malignant tumour? And that process is called neoplasia. Now, there are loads of other terms that you can use besides neoplasia for this process by which a normal cell can give rise to many cancerous cells. So let me just write some of those other ones down. So we also have oncogenesis, so this word makes complete sense. Onco means pertaining to cancer, genesis means to create, so this means creation of cancer. We also have carcinogenesis, again carcino means pertaining to cancer, and genesis means to create, so again this means the creation of cancer. And then there is also tumorigenesis. Tumori of course means pertaining to a tumour, and this means to create a tumour. So all of these terms, oncogenesis, carcinogenesis, tumorigenesis, neoplasia, they all refer to the same process, the process by which you start off with a normal cell, and this is going to give rise to a whole population of cancerous cells, and then you've got a cancerous tumour. Okay, so we want to study this process, we want to study this transformation process, this neoplastic transformation process. Before we actually go into the details of the theory of how it actually happens though, I want to say, what is it? I want to spell out, what is it that makes these cells so strange? Cancerous cells are very, very strange cells. That's why they are so deadly. They're not behaving at all like normal cells. And I want to spell out exactly what properties these cells have that make them so dangerous and that mean that this is such a terrible disease that can kill people. Okay, and these properties that cancerous cells have that other cells don't have are often called the hallmarks of cancer. So that's what I want to start by studying. I want to start by studying what are the special properties that these cancerous cells have, and then we can spell out how do you start off with a normal cell and end up with a cell that has these hallmarks of cancer, or end up with cells that have these hallmarks of cancer. So we are going to study the hallmarks of cancer. Okay, so I'm going to list 6 of these. There are more than 6, but I'm going to list the major ones, the ones that I think, and it's not just me who thinks this, but many people think are the most important ones. So we're going to go through 6 of them. So let's begin with 1 and 2, which seems sensible to do. And 1 and 2 are very much so related to one another. They're 2 sides of the same coin, effectively. So, what is the most famous property of cancerous cells? If you had to say, give me a property of cancerous cells, there is one property that everyone gets first of all, which is that they overdivide. They divide far too rapidly, and that is of course why they form a tumour, why they form a great big lump, because they're dividing too fast and therefore they give rise to lots of cells, and this gives rise to a lump. So, the first 2 hallmarks of cancer, which are the properties of the individual cells, are the properties that are going to give rise to them over-proliferating. So to understand this, we need to understand what controls whether a cell divides or not. So, inside a cell, there are lots of intracellular signaling pathways always active, which control whether the cell is going to divide or whether it's not going to divide. And some of these signaling pathways are activatory for growth or for proliferation. So I'll put these on this picture here. So we'll have these in purple here. So there are some intracellular signaling pathways that are growth stimulatory, and by growth I mean proliferating. So growth stimulatory. So these are signaling pathways that will activate the cell to divide. And on the other hand, there are signaling pathways that stop a cell from dividing. So there are also lots of intracellular signaling pathways inside a normal cell that are inhibiting the cell from dividing. And if the cell is not dividing at present, that is because the two different types of signaling pathways are in balance. So the growth stimulatory pathways and the growth inhibitory pathways are completely in balance, and neither of them is winning. It's a tug of war where it's a complete draw, nothing's moving. Okay, they're both balanced, and therefore the cell doesn't divide. To get the cell to divide, you have to tip the balance. So you either need to increase the growth stimulatory signals or decrease the growth inhibitory signals so that they come out of balance, and overall the cell is then stimulated to divide. So the first and second hallmarks of cancer are going to be about, the first one's going to be about overstimulation of the growth stimulatory pathways, and the second one is going to be about understimulation of the growth inhibitory pathways. So let's put this on then. So we'll start off with hallmark number 1 here. So hallmark number 1 is that the growth stimulatory signalling pathways inside cancerous cells are broken, and they're completely overactive. So overactive growth stimulatory pathways, or growth stimulatory signals, or growth stimulatory signaling pathways if you wanted to do it in full. So growth, whoops, missed out a word, I'll put it in over here. Growth stimulatory signaling pathways. Okay, so something is going to happen. Mutations are going to happen to proteins in these growth stimulatory signaling pathways that it's going to lead to them becoming overactive. And I'm just going to give you another piece of terminology which will become useful later on. In these growth stimulatory signaling pathways inside cells, there are loads of proteins that are involved in those growth stimulatory pathways. And you know, if you've ever studied the RAS, RAF, MEK, ERK pathway, you've seen these proteins, and also the PI3 kinase AKT mTOR pathway. That's another major growth stimulatory signaling pathway. All of those molecules in those intracellular signaling pathways, they're proteins. And those proteins have a name because they are involved in growth stimulatory pathways. They're often called proto-oncogenes. So proteins involved in stimulating a cell to divide are called proto-oncogenes, and if you were to get a mutation in one of the proto-oncogenes genes in the DNA, in the genome, that led to that protein gaining function, i.e. becoming overactive in some way and overstimulating the downstream elements of the signaling pathway, it's then referred to as an oncogene. So proto-oncogenes can transform into oncogenes with a gain of function mutation of some form. So GOF stands for gain of function here. So oncogenes, this word of course is meaning that these proteins are involved in cancer. Proto-oncogenes means the protein before it's actually over-functioning, before it's got the gain of function mutation. It's called a proto-oncogene. It is involved in stimulating cells to divide, but the proto-oncogene is involved in the normal physiology of stimulating cells to divide. Once it's got a gain of function mutation in, it's then involved in actually promoting this cell having cancerous properties, having overactive growth stimulatory signaling pathways, and therefore it's now referred to as an oncogene. Okay, so that's a bit of terminology here. So Proteins that are involved in growth stimulatory signaling pathways and are involved therefore in stimulating a cell to divide, they are called proto-oncogenes. If they get gain of function mutations in them that lead to them being completely overactive, and this overactivates the growth stimulatory signaling pathways inside the cell, then it's now referred to as an oncogene. Okay, so that is hallmark number 1 of cancer, that you have overactive growth stimulatory signaling pathways due to mutations in proto-oncogenes that have caused them to become oncogenes, and these are going to be gain-of-function mutations, GOF mutations. Okay, so next up, hallmark number 2 is going to be that cancerous cells have underactive growth inhibitory signaling pathways. So underactive, and I'll write this out in full, underactive, so just bear with me, growth inhibitory signaling pathways. So the signaling pathways that normally are active and inhibit a cell from dividing, these are going to be underactive, and of course when you've got overactive growth stimulatory pathways and underactive growth inhibitory signaling pathways, then that's going to mean that the cell is going to overall divide far too rapidly. Okay, so this just covers exactly what I've told you here. So both hallmark number 1 and hallmark number 2 are the explanations for why cancerous cells divide far too rapidly. So another piece of terminology: proteins that are involved in growth inhibitory signaling pathways have a special name as well, and these are called tumour suppressor genes because they are involved in stopping cells having tumour-like properties or cancer-like properties. So these are called tumour suppressor genes. Therefore, in order for a cell to gain cancerous-like hallmarks, it needs to have loss-of-function mutations in the tumour suppressor genes. So cancer cells are going to have gain-of-function mutations in some of their proto-oncogenes, making them oncogenes, and they're going to have loss-of-function LOF mutations in their tumour suppressor genes, which are going to lead to underactivity of the growth inhibitory signaling pathways, and both of these together are going to mean that the cell completely overproliferates. Okay, so that's hallmark number 1 and hallmark number 2 of cancer, and these both give rise to the cell overdividing. Okay, so those are 2 of the properties of cancerous cells. Let's go on to property number 3, and I'll get some new interesting colours. We'll have green, I think, and I'll put it here. So property number 3 is that the cells get resistance to the apoptosis signaling pathway. So resistance to apoptosis is a further hallmark of cancerous cells. So what does this mean? So normal cells have a signaling pathway, have many signaling pathways in fact, that can lead to the cell committing apoptosis, which is cell suicide. Apoptosis is the process whereby a normal cell can initiate a pathway that leads to itself being destroyed, and this is a completely coordinated and ordered pathway. This is a mechanism that all cells have ready to be activated if necessary. Now, what would cause a cell to commit apoptosis? Well, normally, if a cell has very strange behaviour, that can then be activated to commit apoptosis. And why would a cell want to commit apoptosis if it's got strange behaviour? Or why is that advantageous for the whole meta-organism? So remember that we, humans, are a multicellular organism. We're made up of absolutely a huge number of cells. Now, the reason that our cells have this apoptosis pathway built into them is that if one cell starts behaving in a really stupid fashion, then it potentially risks the survival of all the other cells. It risks the survival of the multicellular organism, and that's why these cells have an apoptosis process built into them, so that if they start behaving in a really strange way that risks the lives of all the other cells, which is exactly what cancer cells are. They're really badly behaving cells that risk the life of all the other cells. what can happen is they commit suicide, they destroy themselves, and this is to protect the whole multicellular organism. So the single-celled creature dies to protect the multicellular organism. And of course, by killing itself, it's saving itself because all the other cells are genetically identical to it. So it's kind of a beautiful mechanism. Now, it works all over the place. However, in cancer, these cells have actually become resistant to apoptosis, so it isn't foolproof, basically. It doesn't always work. Some cells can become resistant to the apoptosis process, and therefore, even though they should be committing apoptosis because they've got this aberrant behaviour, because they have broken apoptosis signalling pathways. The signalling pathways have become broken, maybe because you've got loss of function mutations in the proteins involved in those signalling pathways that activate the apoptosis pathway. The cell is no longer going to commit apoptosis. So that's another hallmark of cancer, that they don't commit apoptosis, even though they should. Cells that are behaving as strangely as cancer cells do should commit apoptosis, and therefore one of the properties that they have, their very existence, relies on them having broken apoptosis pathways. Okay, so third property is resistance to apoptosis. Let's now go on to fourth property, and I'm just deciding what colour to go to next. We'll have yellow. And so, fourth property is limitless replicative capacity. So let me explain what this means. So this is all about the Hayflick limit, if you have heard of the Hayflick limit before. So limitless replicative capacity. So if you take a normal cell and stimulate it to divide by chucking growth factors at it. So, if you take a normal cell and you chuck growth factors at it, which are signaling molecules which will activate the growth stimulatory pathways, you can get it to divide, okay? You can get it to divide and divide and divide, but it stops after a while. Even if you keep the growth stimulatory signals there, you continue throwing growth factors at it, eventually the cells will stop dividing. It will produce a massive great population of cells, but eventually they'll all stop dividing. And this is because the original cell that the whole population has been produced by had a limited replicative capacity. It could only divide a certain number of times, and this is what we mean by replicative capacity. It could only divide a certain number of times, and therefore there is a maximum number of cells that you can produce from this normal cell here. And then after that, even if onto all of its progeny you throw more growth factors, they will not divide anymore. And that limited replicative capacity of normal cells is known as the Hayflick limit. Now, it would not do for cancer cells to have a Hayflick limit, because then after a certain amount of division they would stop, and therefore it wouldn't be a problem. The cancer wouldn't be a problem. Therefore, cancer cells have the property that they have no Hayflick limit. They don't have a limited replicative capacity. And of course, if you know anything about the telomeres, and in fact I'll explain the telomeres. So the correlate of the Hayflick limit, why do normal cells have a certain number of times they can divide? Well, it's because of structures known as telomeres, and I'll just I'll stick to yellow actually. So the reason that normal cells have a Hayflick limit is because of structures called telomeres, which are on the ends of all chromosomes. So if I just draw a chromosome quickly here, so let's let these two lines here represent one of the chromosomes in a normal cell. Now, of course, a normal cell will have 46 chromosomes, so this is representing one of them. These two lines are representing the two strands of the DNA. Now, on the end regions of the DNA are regions called telomeres. So let's say this little bit here and this little bit here are the telomeres of the chromosome. Now, when a cell divides, the telomeres shorten. In the two progeny cells, the telomeres on all of the chromosomes will be a little bit shorter. So, let me show this here. So here is a cell dividing into two daughter cells, and now what will happen is all of the telomeres on the ends of all of the chromosomes in these two daughter cells will be one shorter than they were in the original cell here. And therefore, when these cells divide into daughter cells, the process will continue. So this one has now divided into two daughter cells, and let's say this one here has also divided into two daughter cells. Now if we look at this generation of cells here, all of those will have even shorter telomeres. They'll have telomeres that are one shorter than the telomeres of this generation of cells, and two shorter than this generation of cell. Okay, and this process continues on until you get to a certain generation where the telomeres are so short that you cannot go any further, because if you went any further, you'd now lose a bit of DNA from the actual coding region of the chromosome where the genes are. So the thing that you have to understand is when cells divide, all of the chromosomes get slightly shorter, and it's okay when you've got these telomere regions on the ends of the chromosomes because they don't matter and you can lose bits from them. But once you've lost the telomeres, then you'd start losing DNA from the actual coding portion of the chromosome, and therefore it would destroy the cell. So that's why you usually have a limited replicative capacity, because the telomeres are of finite length, and therefore once you've divided a certain number of times, once you've got to a certain generation of cells, the telomeres are too short now to carry on. Now, in cancer cells, something has happened that leads to them being able to extend their telomeres, them being able to ignore the normal Hayflick limit. And usually what happens is they overactivate a certain enzyme called telomerase. So in the genome, there is a gene for this enzyme called telomerase, and this is an important enzyme to have active in certain cells in the body where you need a limitless replicative capacity. It's also an extremely important enzyme in development, so in embryology, when you need cells to be dividing rapidly, you don't want them to have Hayflick limits, and therefore we use this enzyme called telomerase. And this enzyme, the way it works is it adds telomeres on effectively, it extends the telomeres. So whilst division shortens all of the telomeres, this enzyme telomerase will boister them back up. It will lead to them becoming longer again. So therefore, it can extend how many times you can divide. Now in cancerous cells, often the way that you get around having a limited replicative capacity is by activating the expression of this enzyme once again through some sort of mutation, maybe a mutation in the promoter for this that therefore allows the cancerous cell to then start expressing this. And that means that the cancerous cells can divide as much as they like because they have this enzyme to extend their telomeres, and therefore they're not ever going to reach the point where their telomeres are too short for them to be able to divide. Okay, so that's property number 4 of the hallmarks of cancer. So before we go on to property number 5 and 6, let's just summarise where we've got to so far. So, we are trying to understand what properties do cancerous cells have that make them so dangerous, that make them so different from normal cells. And we've seen that the major property that they have is that they over-divide, and that the two things responsible for this are hallmarks number 1 and 2. Hallmark number 1 is that they have overactive growth stimulatory signaling pathways due to gain-of-function mutations in proto-oncogenes, and they're then called oncogenes. And property number 2 is that they have underactive growth inhibitory signaling pathways. Therefore, this is brought about by loss of function of these tumor suppressor genes, which are the proteins involved in the growth inhibitory signaling pathways. We've also seen that they have to be resistant to the apoptosis mechanism, because when they're functioning so aberrantly with all their other strange properties, normally that would cause a cell to kill itself to protect the multicellular organism. So for cancerous cells to exist, they have to have resistance in the apoptosis pathway. They also need to have a limitless replicative capacity, and they often do this by overexpressing telomerase, this enzyme that's important in embryology and important in some tissues in the body where we need rapid cell division. For instance, in stem cells, such as the basal cells that we saw in our squamous stratified epithelium of the ectocervix. Okay, right. So let's continue on now with property number 5. So I'll have this pink colour for property number 5. So 5. So property number 5 is that they have something that we call genetic instability. And this means that the rate at which they accumulate mutations is greater than for a normal cell. So remember, a mutation is a permanent change to the genome of a cell that isn't going to be reversed. Now, mutations are usually very, very rare in normal cells. Now, the reason isn't that DNA damage is very rare. DNA damage is actually very common. DNA is being damaged all the time in many cells all over the body. You know, it's being hit by radiation and all sorts of chemicals might be in the cell that are damaging the DNA. So DNA damage is actually very common. So it is actually quite common that you get the opportunity to have a mutation, a permanent change to the genetic code. However, the reason that mutations are actually quite rare is that usually DNA damage is fixed by repair mechanisms in the cell. So normally, if you've got a normal cell, and I'll go back up to my picture of a normal cell. So in our normal cell up here, if something happens that might damage the DNA and might change a few of the bases in the DNA potentially and change the genetic code, normally what will happen is all sorts of DNA repair mechanisms will be activated to repair that DNA and return it to what it was. And therefore you don't get a permanent change in the genetic code, therefore it's not a mutation. In cancer cells, they have dysfunctional DNA repair mechanisms. They've got loss of function mutations in the proteins that are involved in the DNA repair mechanisms, or loss of function mutations in the signaling proteins that are involved in activating the DNA repair mechanisms in response to DNA damage. And this means that when they get DNA damage, it's far more likely that that will actually lead to a mutation. So genetic instability is the name for the fact that their genome is unstable. They are much more likely to get a mutation than a normal cell, and this is because they have dysfunctional pathways that are responsible for DNA repair in response to DNA damage. And that's a really important property because it accelerates the carcinogenesis process, as we'll see when we come on to the process. Okay, so genetic instability. And then finally, the final hallmark of cancer that I want to discuss is that, well, it's the thing that makes cancerous cells so dangerous. This is absolutely crucial for the cell to be called a cancerous cell. If the cell just has hallmarks 1, 2, 3, 4, and 5, it's not cancer without this final 6th property. To be cancerous, it has to be dangerous. It has to be capable of invading the surrounding tissue. It has to be capable of invading tissue where there are normal cells and destroying that tissue. Okay, so property number 6 is that the cells have the ability to invade the surrounding tissue. So tissue invasion is the final property. They invade surrounding tissue and they destroy the surrounding tissue, and this is what makes them so dangerous. This is what effectively makes them just like having an infection. If you've got bacterial cells amongst your tissue, they go around and destroy the normal human cells. They divide at a fantastic great rate and destroy the surrounding cells. Cancerous cells are just like that. They invade the surrounding tissue and they don't give a damn that these cells are their own, that, you know, they were once human cells and that these are also human cells. They destroy them. Okay, and this is why cancerous cells are no good at all. They're awful things to have in your body because they destroy the surrounding tissue as though they're not part of, you know, as though they've forgotten that they're human cells. They've forgotten that they are working for the good of the multicellular organism because they're no longer working for the good of the multicellular organism. They are destroying the multicellular organism. So that is the 6th hallmark of cancer, and that's absolutely essential. That's what makes it dangerous. That's why it will destroy whichever organ it arises in. Now, there is another thing that I want to add on here which is not necessary for it to be called cancer, which is that the cells can become metastatic. Oh, and firstly, let me just explain why you need, how do you get tissue invasion? So normally, remember, cells are not motile. They sit in a space and they don't move into surrounding tissue. In order to invade a surrounding tissue, the cells need to be able to move. They need to be able to move into the surrounding tissue and then start destroying the surrounding tissue. And that requires some sort of mutations that are not very well understood. We don't understand how cells move very well at all, and we certainly don't understand which proteins you need to get mutations in to allow a cell that was previously not able to move to start moving. However, the process by which cells move is going to be disturbed in the cancerous cells, and that's what allows them to invade tissues. Now, people often put along with tissue invasion tissue metastasis, so invasive and metastatic. However, the cell does not need to be metastatic for it to be called cancer. If it's metastatic, then you will refer to it as metastatic cancer, which is even worse than non-metastatic cancer. Metastatic cancer refers to the fact that the cells can start going into blood vessels and then circulating in the bloodstream and then coming out of the blood vessels in far-off sites and then setting up another tumour there. So just drawing a little picture of this. So if we have our cancerous cell here, okay, metastasis refers to the fact that it can go into blood vessels. It gains the ability to go into blood vessels and then circulate in those blood vessels. So here it comes, circulating in the blood vessel, and then it can go far off at a far off site, and then it can come out of the blood vessel, and then it can establish itself at the far off site, and then set up another tumour in this far off site that can start destroying this other organ that's far, far away. Now, if you've got cancer and the cells are capable of invading surrounding tissue, it does not mean that they are capable of metastasizing necessarily. What you require further strange mutations that allow you to do this process, go into blood vessels, survive in the blood vessels, and then come out of blood vessels. Because, you know, it takes something else besides just being able to move in order to be able to go into a blood vessel and then come out of a blood vessel as well. So we do think that you need another set of mutations in order for the cell to become metastatic over just being invasive. And you don't need to be metastatic in order to be classified as a cancerous cell. You just need to be invasive. If you are metastatic, then you are a metastatic cancer cell. And of course, metastasis is one of the major things that ends up killing people with cancer, because the cells start going to the brain and destroying the brain, and that leads to death. Okay, right, so those are the hallmarks of cancer then. I think what we'll do now is we'll have a break here, and then in the next video we'll go over the carcinogenesis process. So we'll actually spell out how do you start off with a normal cell and then get to cancerous cells that have these properties. We'll also talk about the staging of cancer, and this will make the fact that you don't need to be metastatic to be classified as cancer more clear. So we'll do that in the next video.